Cure, Connect, Transform. There is no greater embodiment of Mayo Clinic's plans to revolutionize healthcare than the Center for Individualized Medicine. We discover, translate, and apply new individualized medicine advances in patient care. Using genomics and multiomics could lead to vast advancements in knowledge of the causes and risk factors for many diseases. Our researchers and clinicians work together to accelerate discoveries in 4 strategic areas. Simplify and scale. In Genomics in Action, we work to transform the practice by providing the knowledge, tests, and tools needed to make it easier for clinicians to integrate genomics into routine patient care. Individualizing cures with genes, bugs, and drugs. Cutting-edge therapies being developed with high-definition therapeutics are driving precision medicine science beyond diagnostics to provide personalized cures. Connecting data to practice. Our exciting work with Omics Data Platform is centralizing genomic and multiomic data into a single repository, which will allow for the most individualized medical approaches possible. A multi-omics future. In Beyond DNA, we are advancing patient care by using molecular information from the microbiome, epigenome, exposome, and more.
The Center for Individualized Medicine and Mayo Clinic are in a unique position to fuel new discoveries to improve care for patients powered by sophisticated research and artificial intelligence. From providing the best individualized care to addressing the world's most challenging healthcare problems, Mayo researchers here are relentlessly pursuing discoveries that deliver hope and better health to people today and for generations to come. Thank you.
Good afternoon. My name is William Palmer. I'm the Associate Director for Education for the Center for Individualized Medicine in Florida. I'd like to welcome you all to our Florida Center for Individualized Medicine Grand Rounds. Our Grand Rounds lecture series is designed to highlight the latest in scientific discovery and innovation at Mayo Clinic and demonstrate how individualized medicine is being translated into practice to meet the current and future needs of our patients. It's my pleasure to introduce today's speaker, Dr. Liewei Wang. Dr. Wang is the Bernard and Edith Waterman Director of Pharmacogenomics Program for Center for Individualized Medicine. She's also a consultant in the Department of Molecular Pharmacology and Experimental Therapeutics at Mayo Clinic and is Professor of Pharmacology in the Mayo Clinic College of Medicine. The research of Dr. Wang focuses on pharmacogenomics, a critical component of individualized medicine that studies the role of inheritance in variation in drug response. Patients treated with the same dose or regimen of a drug can display very different responses, ranging from lack of efficacy to severe toxicity. Dr. Wang's laboratory uses various high-throughput omics tools, including genomics, transcriptomics, and metabolomics, to identify biomarkers that can help predict clinical response. At the same time, her lab uses various models, including cell and patient-derived xenografts for patient samples, to validate and understand the biology of these biomarkers. Throughout this talk, please use the Q&A function in Zoom to submit your questions. We have some time at the end for Q&A. For those of you who have not yet claimed CE credit, the code in all caps will be displayed again and posted. It's PGx-2022. PGx-2022 in all caps, and I'll be posting that in the message chat shortly. So without further ado, I present Dr. Wang and her presentation, Pharmacogenomics, Discovery and Translation. Dr. Wang.
Thank you so much. I share my screen. Thanks for the kind introduction, and thanks for inviting me, give me this opportunity to present some of the latest work that we've been doing. My laboratory is really focused on oncology precision medicine. We focus on both the somatic genome as well as the germline genome. Here, I mainly describe the application of some of the functional genomics to characterize the germline SNPs that are relevant in pharmacogenomics. So as you already heard, probably most of you know the definition of pharmacogenomics is try to refresh your memory. This is a study of the role of inheritance in individual variation in drug response phenotypes ranging from lack of efficacy to severe toxicity. Obviously, Nowadays, when we talk about these, it's really expanded into multiple omics, not only just the DNA, but also information RNA, proteins, could all use as a biomarker to predict response. It is a critical component of personalized or individualized medicine. What is our clinical goal? Why we want to perform pharmacogenomics? The first is do no harm. We want to avoid adverse drug reactions. Second is really want to maximize the drug efficacy, particularly in some area like oncology, use those biomarkers specifically for selection of the appropriate patients for different treatment. So this is really our ultimate clinical goal using this information. At the scientific level, what is the basic science we're doing? We try to link the variation in genotype to variation in phenotype, but also determine mechanisms that underline this relationship. And eventually, by understanding the mechanism, we can translate into the link into our enhanced understanding of treatment as well as the prevention of the disease. So when I talk about pharmacogenomics, probably most of you think about, very well-known genes, particular genes in drug metabolism, enzyme transporters, some of the drug targets, and coding SNPs, those SNPs that are well-known in these genes. A lot of them already show clinical utility. Mayo also is, like many other places, have already started implementing these common genetic variation in these well-known pharmacogenes as a tool to help a physician when they prescribe drugs that might have pharmacogenomic implication. However, with the increasing technology like sequencing, we also know we are discovering more and more common and rare SNPs in these genes. Challenge is many of those SNPs of very unknown significance. How are we going to characterize these SNPs functionally? So I will touch briefly on that using high throughput functional genomics, the technology that we have in the laboratory. We also understand that 98% of the genome is non-coding. So many of those SNPs are in the regulatory regions. So how they impact the function is also a bigger topic. And in some way, it's also a challenge how we study these SNPs in the regulatory region, try to understand how they might link to the genes, inference gene transcription or gene function. The other is we know drug response is a complex phenotype. The current knowledge with some of those genes probably only explain a small variation that we observed in the clinic. So there are a lot of information in genes that are beyond the current, that are well-known pharmacogenes. And SNPs in those genes, such as genes encoding transcription factor that can regulate pharmacogenes or pharmacogenomic genes, could also contribute to ultimate drug response. I will touch one example, actually showing SNPs in transcription factor regulating these well-known pharmacogenes could also impact drug response. Lastly, I just want to briefly touch using our most recent example, really to illustrate the complexity. The genotype of the individual SNP impact a function like a gene expression is not only rely on the genotype of the host genome, but also depend on the presence of different chemical compounds, either endogenous hormone or exogenous chemical compound like a drug. So in other words, there is a SNP genotype drug interaction together influence gene regulation. As I said, most of these are well-known pharmacogenes that has been implemented from the implementation perspectives. It's a reactive process, and most of these SNPs are well-known. These are genotype-based clinical assay. So in a way that physician, this is reacted to the physician's prescription and then trigger genotyping of those SNPs to give the physician information. Obviously, many of these genes that has been well implemented, well-known has been implemented across Mayo all three sites. This is the list that I just showed the Mayo PGx drug gene pair, you can see a lot of these SNPs that concentrate in 13 pharmacogenes, including a lot of these cytochrome P450, like a 2C9, 2C19, 2D6, as well as some of the phase 2 drug metabolism enzyme like a TPMT, TSPYL. And those information that is implemented, that are being there, like I said, it's really reactive. Physician prescribe a drug that have these pharmacogenetic indication, then we trick the alerts to alert the physician to get the genotype information that eventually, hopefully, can facilitate physician decision on therapeutics. So what we have thought all past couple years really is, can we really bring this reactive into a preemptive setting instead of using genotype those known, rely on sequencing with the increasing availability of sequencing technology, really to identify also those that we don't know currently that might also have functional impact to drug response. So with that in mind, within Center for Individualized Medicine led by Pharmacogenomics program, together in collaboration with the Baylor Genome Center, we designed this Mayo Right 10K study. The study is being published already early this year in Genetic Medicine. So the briefly describing of the study is basically Baylor created a 77 pharmacogene sequencing panel that allow us to sequence 10,000 Mayo biobank DNA subject. Obviously, these 10,000 Mayo patients all receive care. Mayo, we have EHR going back 20 years, which allow us to perform association study in a retrospective fashion, but also because these are Mayo patients, they keep coming back. So we also can have perspective view if we have these preemptive genotype already in the EHR, what this might help and how this can help a physician prospectively going forward. So the characteristics of these 10,000 patients, you can see here on the table, pretty evenly split between female and the male, a little bit more for female. We also try to enrich the patients with those older than 50 years old, with the assumption that older patients are likely to have polypharmacy, so pharmacogenomics information in the EHR in the future might be more helpful. And you can see on the side, our consenting rate actually is pretty high. Obviously, these patients are already being consented through the biobank for the application or utilization of their DNA. We send the right 10K letter just to get their agreement for specific participation in this particular study. Here's the table, show the 77 genes that are built through the Baylor Genome Center. I highlight these 13 genes that are already being implemented at Mayo, which is the table I showed before. Previous slides, the 13 genes count 21 gene drug pair. If we look at in terms of at the population level, very consistent with our previous smaller study, among these 10,000 patients, 99% of these patients have at least one actionable variant in these 13 genes that we already implemented. So in other words, this is the pharmacogenetics is truly for everyone. And these information in the EHR potentially have a wide implication for most of those patients. And interestingly, when we look at these novel variants, because remember here we use the sequencing with the assumption that the sequencing will give us more information, and not just more SNPs information, but also potentially functional SNPs. So here is just looking at these 13 genes, excluding these actionable variants that the Mayo lab already genotyped. On average, among these 10,000 patients, we have addition of over 73,000 predicted deleterious SNPs. And on average per sample, each patient might have 7 additional deleterious SNPs. So that does show sequencing gives us way more additional information. And obviously, some of these SNPs are rare, but if you aggregate all these information together at a population level, it's still a substantial amount of contribution that we should account in the future when we are doing assessment in the clinical side. As I mentioned earlier on, one of the challenges is even those are coding SNPs, how we assess these SNP function in a relatively high throughput manner, given the fact that now that we have identifying hundreds of new variants, for every gene using the sequencing approach. So in the laboratory, we have these create a high throughput methodology that allow us to really assess coding SNPs in a relatively high throughput fashion. Here, just taking 2 genes, 2C9, 2C19, these are 2 very well-known cytochrome P450 genes, and there are coding SNPs that are being genotyped routinely, showed here on this figure for 2C19. This particular nucleotide at the position 1076, as well as this particular nucleotide at the position 1349 in 2C19. These are the 2 mostly commonly genotyped SNPs, which result in null function or deleterious. These are the deleterious SNP result in null function of the CYP2C9 and the 2C19. So when we put all the rest of these novel SNPs in the context of these 2 well-known pharmacogenetics variants, and we use the high throughput methodology, assayed over 100 SNPs to select based on the minor allele frequency. You can see here, some of these are relatively rare, but when you look at the function, about 20% of the new variants in CYP2C9, or about 30% of these new variants in 2C19 actually have severe deleterious function, as I said, if you put in the context of these 2 well-known pharmacogenetic SNPs in these 2 genes. The high throughput methodology is really taking a fluorescence based assay with the concept that most of those coding SNPs could impact function of the gene through decrease its protein level. So the lower protein, the lower fluorescence signal we obtain. So this is basically a base for this high throughput assay. So that really tells us sequencing give us or provides us many additional potential functional variants that even though at the individual SNP level, they might be rare, but when you aggregate at a population level, that's still a substantial contribution. You can see 20-30% among all the variants we assayed actually are functional. So we are talking with our clinical lab here, thinking about how we future going forward, how we can implement these rare but functional SNPs in the clinic. So this is just one example that showing how we can really go beyond the current SNPs, coding SNPs in these well-known cytochrome P450. As I alluded earlier on, drug response is complex. SNPs in these genes themselves only contribute a limited amount of impact. But how we can start really looking beyond these pharmacogenes and genes that might regulate these CYP or cytochrome P450. So I'm showing here another example really to illustrate transcriptional factor that regulate cytochrome P450 or SNPs in these transcriptional factor regulating cytochrome P450 could have also substantial impact on clinical drug responses. So this is really go beyond SNPs in these cytochrome P450 gene themselves and try to aggregate additional genetic information. So the transcription factor that I'm going to talk about, these family, is actually coming from our early GWAS association studies, where we start identifying this family of transcription factor proteins and realizing they might be a major family regulating multiple cytochrome P450. So this is the early GWAS study performed about 10 years ago. And really, the question at that time we asked is, can we identify genetic variation contribute to baseline estrogen level in 800 breast cancer patients? Because we know estrogen contribute is a risk factor for ER-positive breast cancer. So here we just ask, are there any genetic variants through contributing to the baseline estrogen level might be related to breast cancer risk? So we did a GWAS of baseline estrogen level measured by mass spec. And here is the GWAS results. Each of these are indicating a SNP aligned based on their chromosome localization. And the y-axis is the p-value, how significant each of these SNP associated with the phenotype. In this case, level for estrogen in these 800 women. So one of the top signals showed up, I highlighted here, a SNP on chromosome 18, close to a gene called the TSPYL5. And zooming in, there are a series of SNPs that are highly linked in this region that are within this TSPYL5 region. So what we further demonstrated, a long story short, is that TSPYL5 is a transcription factor, or at least a transcription co-regulate that can specifically regulate CYP19, which is the enzyme encoded enzyme aromatase. Aromatase limits step during the estrogen synthesis. Remember, our phenotype is estrogen level. So we focus on the rate-limiting enzyme that are converting precursor to estrogen. So that's why we first look at how this particular gene might regulate cytochrome P450-19A. Here, I just briefly show when we overexpress TSPYL5, or when we knock down TSPYL5, the cytochrome P450-19, the CYP19, accordingly going up or going down. So that does tell us there is a relationship between these two genes. And we do have CHIP assay to show that TSPYL5 bind to the promoter region of CYP19, specifically promoter region encoding promoter region that is responsible for CYP19 expression in adipose tissue, in breast tissue. So this is all very relevant in a way that this particular new family of proteins potentially can function as a transcription regulator for cytochrome P450, in this case, CYP19. And what we follow up with the initial observation is really expanded to ask a much broader question, whether this particular family of transcriptional factor, which have, you can see 6 different family members, can it regulate at a much broader sense many cytochrome P450 genes? So this family have 6 genes, including the 5 that I was just described that relate to estrogen synthesis by regulating CYP19. And TSPYL3 is a pseudogene, but 1 to 4 are the 3 genes expressing almost all tissue. So we really follow up a study or focus on these 3 genes. And for cytochrome P450, many of you are familiar with, has 57 different CYPs. and also comprise drug metabolism enzymes, which mainly categorized into CYP2, 1, 2, 3 subfamily. And also, these CYPs also contain a lot of hormonal metabolism enzyme or synthesis enzyme, including CYP17A, CYP19. CYP19, as I alluded, it's a target for a lot of drugs, including a class of drug called aromatase inhibitors that are commonly used in the treatment of ER-positive breast cancer. And CYP17 is equivalent of CYP19, but this is a rate-limiting enzyme responsible for synthesis of testosterone and androgen in the body. So it's a male hormone. And there's also drugs specifically targeting CYP17A called Abiraterone, which is now standard of care for prostate cancer. So when we start looking at these enzymes, the transcription factor family, and the broader range of cytochrome P450, what we found, I'm not showing you the data, but in our published paper, we do find a series of cytochrome P450, including 2C9, 2C19, CYP3A4, CYP17, all regulated by TSPYL 1, 2, and 4. So there is a general kind of phenomenon between these two family of proteins. What I'm going to show is just one particular study focused on the CYP17 and 3A, as well as their relationship to TSPYL family member. And SNPs in the transcription factor can really impact drug response, in this case, particular Abiraterone, the drug target CYP17. So in prostate cancer, here are the two prostate cancer. What we did is we knock down these TSPYL family protein 1 to 4. These are the three more ubiquitously expressed in multiple different tissues, including liver, prostate. So we knock down these three family and then assess specifically, in this case, CYP3A4 and the CYP17A, because as I said, the CYP17A is a target for the drug Abiraterone, and CYP3A4 is a major metabolism enzyme for Abiraterone. So sort of we have the two sides of the drug all covered. And what we find is that knock down these three transcription factors, we do see upregulation of CYP3A4, but downregulation of CYP1. So there is an opposite relationship in terms of regulation. And when we overexpress, it shows, the opposite results compare when we knock down these three transcription factor. And similar observation was seen in another prostate cancer cell line. So in other words, overexpression or these genes can activate or regulate in a positive way of this target CYP17, but transcriptionally suppress the level of CYP3A4, which is drug metabolism enzyme for Abiraterone. And this also translates into the function, specifically when we look at the CYP17. So CYP17, as I said, is a rate-limiting step enzyme to generate male hormone and androgen or testosterone. And these are the driving force for prostate cancer cell growth. So here we just did a simple experiments knocking down these three transcription factors, or combine the knocking down all of these transcription factors, and measure the downstream molecules like DHEL or PSA. These are indication of activation of androgen pathways. We also assess the cell proliferation. So indeed, when you knock down TSPYL, it sort of down-regulate CYP17A. And consequently, it downregulates the androgen-dependent regulation of DHEA and the PSA. So you do see these levels all go down. And also because this downregulate androgen synthesis by downregulating the enzyme that produce androgen, you also see cell proliferation decrease because, as I already said, androgen is the major driving force for prostate cancer cell growth. If you cut off that driving force, cell proliferation decrease. So all of these really confirm that indeed the class of transcription factor that we identified that regulate cytochrome P450, in this case very specifically the CYP17A, can translate its impact on the downstream androgen-dependent function. So we then ask the question, come back to genetics, pharmacogenetics. Can we identify through the public database all these coding SNPs in these transcription factor with the assumption that if those SNPs influence the transcription factor function, they might in turn influence the cytochrome P450 and could have an impact on response to certain drug. So we use the same Abiraterone as our sort of a study case. And in this case, we do identify a series of coding SNPs with a minor allele frequency over 10%. In 1 and 4, we didn't find any high frequency coding SNPs in TSPYL2. So here are the list of those coding SNPs with amino acid change. And when we specifically look at these SNP function on CYP3A4 or 17A expression, we compare with the wild type when we overexpress all these variant SNP. What you can see is I highlight this. The only one really show a dramatic functional change is this proline to serum change at the 62 amino acid level in TSPYL1. In this case, when you overexpress this variant SNP, it abolished the ability of TSPYL inhibition of CYP3A, 3A4 transcription. It doesn't have any effect on CYP17A. So in other words, this SNP has some selectivity on the downstream substrate. So with that in mind, if you have this variant SNP, compare with the wild type, it actually removes the inhibition of the transcription factor. In other words, this transcription factor really suppresses CYP3A4 transcription. And if you have this SNP, you remove this suppression effect, actually increase the expression of CYP3A4. So in other words, when you have more CYP3A4, it metabolizes the parental drug Abiraterone much faster. So when we measure the parental drug level Abiraterone in cells, we found the phenomenon fit with our hypothesis. So compared with the wild type overexpress of this variant SNP, when we measure Abiraterone, the level dramatically decreased. And we also test the effect of those SNPs on the treatment of cell, cell growth. So in this case, if the cell has this particular variant, and in other words, it has a lower parental Abiraterone drug, which is the active drug, it actually has a worse effect on the inhibition of cell proliferation because you don't have enough drug to inhibit cell proliferation. So this is truly the case. If you look at this black line is the variant SNP, and here in 2 different cell lines. So if cell overexpresses this variant SNP treated with Abiraterone, it actually grow faster than if they overexpress this wild type. So in other words, the variant cause increased CYP3A4, which enhance the metabolism of the parental drug, reduce the active drug, and that can lead in less therapeutic effect. And we also assess this relationship in our patient data. This is a sponsored prostate cancer prospective study called PROMOTE, where we recruited 90 patients. And we also have DNA, tumor DNA, germline DNA all collected. So here are the genotyping results in relationship to time to progression. The blue line are all 6 patients among these 90 patients who are homozygous for this TSPYL1 SNP. And all of them have a very short progression-free survival time compared with wild type or even heterozygous. So this is truly a deleterious SNP that can significantly impact patient outcome. So here's a model. What we think is normally Abiraterone, which is a drug used to inhibit CYP17, to inhibit androgen synthesis or testosterone synthesis, which can obviously drive down the risk of prostate cancer, reduce the prostate cancer cell growth. And if we have this SNP in a transcription factor, TSPYL, that normally suppress the expression of CYP3A4. But if you have this SNP, enhance the expression of CYP3A4, which dramatically increase the metabolism rate. So Abiraterone then can be excreted much faster than if patients have the wild type TSPYL SNP. And that could cause the clinical impact that I just showed you in the previous slides, which those patients, even though they treated with the standard dose, but probably doesn't get the right amount of exposure concentration. That's why their outcome is poor. Even though this enzyme, this transcription factor also regulate the target, but specifically for this genetic variant, actually doesn't have any impact on the target expression. So there is a selectivity towards the regulation of CYP3A4 in this case. So this is another example, really, it kind of forces us to really think about identifying those upstream transcription factor or other regulator of those well-known pharmacogenes and really aggregate information together in the future that can help us better predict a drug response. The last example really is focusing on a little bit complicated. The scenario is that we not only consider the SNP genotype per se, but also put this information in the context of a different individual drug. So there is a SNP genotype drug interaction together influence gene function. So we know that approximately 90% of those GWAS signals map outside of the protein coding region. Challenge is most of these GWAS signals that we really don't have any functional indication, don't know how they behave or why they associate with the phenotype. So most of these SNPs in the regulatory region, we hypothesize that these SNPs probably influence functions through influence gene expression regulation. So so-called expression quantitative trait loci. And there are public database like a GTEx portal, which really give all the information on cell or tissue specific SNP gene expression relationship. However, most of these relationships are characterized mainly at baseline, in other words, without any challenge or in the presence of chemical compound. But at the same time, we also know this relationship are affected by a variety of factors. I already alluded, there is cell tissue specific SNP gene expression relationship, but there's sex bias gene relationship, SNP gene relationship, but also drug or hormone or chemical compound modulated SNP gene relationship. We call these actually Pharmaco or PGx-eQTL relationship. So if I just say simply, if you look at these PGx-eQTL, by which I mean those SNP gene expression relationship occurred in the presence of the drug. Here, just two very simple examples to illustrate that. The y-axis here shows the gene expression. X-axis here is a different genotype represented by 3-bar Y-type homozygous, heterozygous, so homozygous of the variant SNPs. So if this relationship occur at the baseline and doesn't change post the drug treatment, and we don't consider these as PGx relationship. But if we use in the presence of drug, either reappear or disappear or change the direction of those genotype gene expression relationship, we call those PGx-eQTL. So this is a sort of give you some very simple schematic description of what we call PGx-eQTL. This concept actually initiated from studies we've done over the years. One of the early study lead us to have this discovery is a GWAS study we performed about 10 years ago that was published in Cancer Discovery at that time. In this particular study, we really is try to identify genetic variation that relate to tamoxifen prevention outcome. Tamoxifen is the chemotherapy that actually approved by FDA to prevent breast cancer in high-risk women because of its rare but very severe side effects. So the utility of this drug or this class of drug in clinic as a prevention agent is not popular at all. Besides, statistically, you need to treat 8 women to prevent 1 case. So we had that hypothesis that can we utilize clinical trial, prevention trial to identify genetic biomarker that can help us to predict and select patients who are best suited for the prevention treatment. So this is basically a case control GWAS. We have access to 30,000 women enrolled in the 2 largest prevention study performed in North America, P1, P2. These are the 2 studies led FDA approval of tamoxifen or this class of drug in prevention setting. Despite of the 5-year prevention treatment, there are still close to 600 women develop invasive carcinoma. So we take these 600 cases paired with 1,011 or 1,100 controls and did a GWAS. And this is the GWAS, as I said, it's a Manhattan plot display the GWAS association results. The top signal shown here is the SNP in the gene called the ZNF423, zinc finger protein 423. Based on the name, we assume it is a transcription factor. However, we don't know what these SNPs really do and how they might link to this ZNF423 gene and how this relationship link to the drug effect, in this case, tamoxifen, which is the estrogen receptor modulator. As well as the clinical phenotype, in this case, is a breast cancer risk. I want to say, even though the GWAS was only about 10 to the -6, which really not statistically meet the GWAS significance, we still decide to pursue functionally because this is the largest sample sets we can have to get these results. So in order to really elucidate all these relationships, we have to have an in vitro cell system that we can manipulate. So here we have come up in the lab a 300 cell line. These are lymphoblastoid cell line immortalized from 300 individuals. You can see from 3 ethnicity groups too. And these obviously, like our mini in vitro pharmacogenomic panel, represent common genetic variation among individuals. We also have GWAS expressing all these data in each of these cell lines that allow us to pick individual cell line with specific genotype of, in this case, ZNF423 SNP. So we pick A cell lines with the Y type, A cell line heterozygous, and A cell line homozygous variant. And then we can manipulate the cell line to mimic the clinical setting So we treat these cell line with the physiological estrogen level because these women, have endogenous estrogen level. Then hit with tamoxifen, just like in the clinical setting. And interestingly, if you see this graph here, the Y type is the red line, and you see in the presence of estrogen, Y type upregulates, but the variant sort of stay, or the heterozygous stay lower. However, when we treat with tamoxifen, you see the flip situation. The variant actually start upregulate versus the Y type keep dropping. So there is, like I said, both the genotype and the drug effect on gene regulation. So when we here we show the SNP indeed have relationship with the nearby gene ZNF423, which is transcription factor. But how we link this gene with the breast cancer risk? So we go to TCGA, all the breast cancer data, simply did a correlation of this particular gene with the rest of the genome, with the assumption that genes highly correlate with this transcription factor might actually regulate by the transcription factor. And we only focus on those genes that are well-known breast cancer risk. And long story short, one of the top significant hit is a well-known breast cancer risk gene, BRCA1. So we did additional work really confirm ZNF423 is a transcription factor for BRCA1. So here we knock down or overexpress ZNF423. We see BRCA1 also downregulate or overexpress. Here is the CHIP assay. We do see ZNF423 directly bind to the promote of BRCA1. So now we link the ZNF423 to the gene BRCA1. And when you look at this phenomenon, a SNP drug-dependent regulation, and this really translates into the BRCA1. What we found as association is this variant SNP is a protective SNP. In other words, a patient with this variant SNP actually have decreased risk to develop a breast cancer. To some extent, we think because of those patients start with a low BRCA level, but in the presence of tamoxifen, these variant patients actually have upregulation of BRCA1, which you need normal BRCA1 to really function to protect you have breast cancer. So that's the initial observation. Indeed, there is a SNP drug interaction in terms of regulation, and this can translate significantly into the downstream function. So here is just some of the summary I already alluded in terms of our observation. And importantly, I want to point out is this is basically an additional variation contributing to patients without BRCA1 mutations. We all understand the BRCA1 mutations, the BRCA null situation. But here is a very common with a allele frequency about 35 to 40%. It's a very common SNP, not really in the BRCA1, not even in the BRCA1 region, but in a transcription factor that regulate BRCA1, which can substantially contribute to downstream phenotype. And on the other hand, this SNP could also have implication for response to other treatments, such as PARP inhibitor, because we know BRCA null setting they are sensitive to PARP inhibitor. So here I just show one figure from our follow-up study to illustrate this point. If you look at these scenarios, so I graphed on the left side how this SNP and the drug my interaction with the DNA region and regulate ZNF423 transcription, in turn regulate BRCA1. So if you focus on the lower two scenario, if patients have the variant SNPs and they cannot really have a full activation of ZNF423 because of the potential, might be, that's our hypothesis, might be because of the transcription factor or co-regulate, they don't bind very well on the DNA because of this SNP in the presence of natural ligand estrogen. And this can cause the downregulation back. But on the other hand, if here is a Y type SNP, only in the presence of tamoxifen, in this case, you will have this potential scenario and reduce expression of ZNF423, which can downregulate BRCA1. And in those two settings, we also see enhanced sensitivity to PARP inhibitor. So here's the cell line xenograft. We created a CRISPR knocking breast cancer cell line. So the wild type and the variant cells that have exactly the same background, except this particular SNP difference. And if you look at these treatment response, and if the tumor has the variant SNP, they are natural sensitive to olaparib, which is a PARP inhibitor. But if you have this variant, or you have this wild type genotype, and in this case, these these tumors respond much better to tamoxifen plus olaparib, because you can see only under the tamoxifen or in the presence of tamoxifen, you will create this phenocopy of BRCA null, which can then sensitize to olaparib. So all of these really indicating SNPs and drugs can both influence gene regulation, which have substantial downstream impact, and not only for disease risk, but also potentially for treatment response. So I'm very quickly going to move to a very recent publication, just published last month in Nucleic Acid Research. This is in collaboration with Dr. Winchelbaum as well as the Aldocs lab. And this is really to expand these anecdotal example that I just show you into genome-wide. And in this case, we focus on agonist or antagonist targeting glucocorticoid receptor. The reason we expanded to a different receptor system is we really want to show this SNP drug-dependent phenomenon is generalizable. So here is the study design, really taking advantage of the lymphoblastoid cell line I talked about. Treat these cell lines 30. We selected 30 lymphoblastoid cell line treated with cortisone, which is an agonist, just like estrogen, or antagonist C297, just like tamoxifen, or combined it together. And identifying, and then we did RNA-seq and did a SNP gene expression relationship analysis to identify PGx-eQTL SNP for both cortisone specific as well as for C297 specific. And then take those SNPs using high throughput functional analysis called the STAR-seq, together with epigenomics like a high chip, to really characterize these SNPs functionally, and take those functional SNP, ask the question, do they have clinical impact, by determining their association with the public available GWAS association results. So this is just a very briefly, I show some of the results. This is one figure that integrate all the results in the presence of cortisone treatment. So the three layer, the out layer is represent the genotype of Y type. Middle layer is the heterozygous. The lower layer, the inner layer is the homozygous variant SNP. And this is basically to indicating a SNP gene relationship. You can clearly see from lower expression, which is the blue, all the way to high expression, there are correlation between different genotype and expression level. So that means indeed that those genetic variants does have impact on expression and in the presence of cortisone. And a lot of these, I'm not going through every one of these data, and these dots just indicating all the linked SNPs around these region. And then we also have these lines indicating in the genome or on the chromosome, these are glucocorticoid receptor or GR binding site. So here are the gene names. These are the genes surrounding these SNP area, and highlight those genes are the genes that we actually identify through the GWAS results. So in other words, those SNPs close to these gene area showed up in top significant GWAS signals. So overall, we identify about 200 cortisone dependent or C297 dependent PGx SNP EQTL relationship. And here, just showing one of the high-throughput functional study called the STAR-seq, which allow us to assay all 200 SNPs simultaneously, really to ask a question whether those SNPs are in the region that are transcription active, which is like an enhancer region. So this STAR-seq high-throughput assay is really to assay enhancer activities. So that allow us to identify all the SNPs that potentially have functional impact. So among these 200 SNPs that I just listed, identifying through the initial analysis, by performing STAR-seq analysis, we confirm close to 70%. You can see the number shown here, 102 cortisone dependent or 32 C297 dependent SNPs are really functional validated. In other words, those SNPs in the region of transcription active region that truly can impact gene expression. And when we take all these functionally validated SNPs, then say, are there any of those SNPs truly linked to the phenotype that the people have already tested through various GWAS? So here are public GWAS information we can get from the GWAS catalog or UK Biobank. And indeed, 20% of those SNPs do show as top signal with a variety of phenotype people have already performed the GWAS on. And many of those actually very relevant to glucocorticoid biology. You can see inflammation, immunity, psychiatry, cancer, osteoporosis, metabolism. So that really indicating through this approach, a lot of these GWAS signal in the regulatory region that currently we don't understand how they function. Probably we didn't look at the environmental factor or other factors such as endogenous chemical compounds or exogenous compounds like drugs they expose. So in summary, we do feel this type of PGx-eQTL have significant functional impact, which has been really not well understood or studied before, and potentially can explain a lot of these clinical GWAS association results. And certainly the observation indicates the inference of SNP and drug effect on various of clinical phenotypes. So just in conclusion, I sort of have several examples really to illustrate the PGx has evolved. These are just all of them are germline SNPs. So PGx has evolved from single drug gene pair to including SNPs in the regulatory region, SNPs in genes beyond the current PGx genes, as well as SNPs that have differential impact based on the presence of different chemical compounds. So high throughput functional genomics definitely adds significant values to our understanding of the SNP contribution, both in vitro as well as potentially in vivo or through clinical studies. Through all of these obviously, a lot of these are discovery, but at the same time, a lot of these discoveries were made from our clinical observation. So future clinical implementation needs to consider all the functional SNPs, which collectively really can better predict a drug response. Obviously, this extends to how we integrate these SNPs, how we build a future algorithm for these SNPs. So I think I stop here. Want to acknowledge a lot of people involved, particularly on the breast cancer side, Dr. Winchell, Dr. Ingo, Dr. Gatz, who have involved in our early GWAS studies, and Ingo and Gatz are the two really help us access a lot of these big clinical GWAS study. And for the PROMOTE study, Dr. Tan from Florida, as well as Ronnie, Dr. Clara for bioinformatics statistical analysis for the right 10K. It's a huge team, and you can look at the authorship, over 100 people from Mayo as well as from Baylor. Obviously, our PGx group within Center for Individualized Medicine particular Leila Jones, who's the administrator for the PGx group, really support all these studies. Our pharmacy colleagues, particularly in the right 10K studies, they contribute substantially from the implementation side, as well as all the postdoc graduate students who led all the studies and the funding, obviously, from SIM, as well as from extramural funding. So I'd like to take any question if I still have some time.
Thank you very much, Dr. Wang. Turn my light on here. One question that I had, just in hearing the context of how quickly a lot of these drugs are certainly evolving too, especially in the oncology space. Is there a plan or your thoughts on integrating how this works in clinical trials? How can we best leverage the technologies and the advances that have come out of your lab and similar labs for drug development to try to get ahead of adverse effects, even preclinically?
Yes, certainly our goal is for a lot of these information eventually, can be translated into the clinical, but at the same time, a lot of these technology as well as sort of conceptually, a lot of these study can be also integrated during prospective clinical trial. You can see some of the studies we actually do use clinical prospectively collected clinical samples to derive information, which we then can later on, to go back to the clinical side to integrate to the next studies using these biomarker. Obviously, it's from the discovery to clinical, it's not as simple as we just talk about. There is obviously a long way that can anything eventually can be implemented on the clinical side, not only from scientific perspective, but also from the clinical utility. There is a lot of needs for validate these findings from the clinical side in terms of the clinical utility.
Fantastic. Well, I want to thank Dr. Wang so much for a superb presentation to hear the cutting edge direction of this science. And I hope everyone has a wonderful holiday season. Don't forget to fill out the CE submission to get your credit. And we will see you next time for another SIM Grand Rounds. Have a great holiday, everyone.
Thank you.